Swayam Prabha. Digital India. Educated India.
In this lecture, we will discuss about the properties which can be derived from the protein 3D structures. In the last class, we discussed about protein 3D structures, right. What did we discuss in the last class? How to get the 3D structures?
X-ray,
experimental X-ray crystallography, NMR spectroscopy and electron microscopy, and there are several novel arrays, right, for understanding the structure and function of globular proteins using X-ray crystallography or NMR spectroscopy, right. So, all the structures which are solved by X-ray or NMR or electron microscopy are deposited in the database, right. What is the name of the database? That is a protein data bank, right. Protein data bank is maintained, right, with a research collaboratory in structural bioinformatics, right. So, there are several mirror sites in Japan, in US and in Europe, right. So, that you can get the access from different parts of the world. So, what are the major contents of protein data bank?
there will be header information.
Right, they have the header with the name and the source, right, and the how the resolution and the secondary structures, publications, and you can have the data on the coordinates, right. So, what are the information you can obtain from the coordinates?
So,
Atom name, atom number, residue name, residue number, chain information, and the coordinates, exhausted coordinates, B factor, occupancy, right. We will get all the information right for all the atoms, right, in a protein. Then we discussed about the visualization tools. There are several tools, right, which are commonly available to view the structures, and you can also manipulate the structures in different directions, and you can calculate the bond length, bond angles, torsion angles, mutations and so on. So, then we discussed in detail about pi mole. So, pi mole has several applications. What are the potential applications of pi mole?
so it calculate the distance.
You can get the different measurements, and you can view the structures, and you can make high quality figures, right, and you can do the mutation analysis, we can do the interactions, whether hydrophobic or electrostatic or different interactions, right. So, it has wide applications, right. So, if you go through the pi mole, right, you check the tutorials and you will get several options you can utilize pi mole effectively. So, we take the protein structures, mainly they are classified into 4 different classes depending upon the secondary structures present in protein structures, right. So, if you what is the secondary structures? The major secondary structures are alpha helices and beta strands, right. How these secondary structures are distributed in 3D structures? Based on that, we classify the proteins into 4 different groups. The major one, that is all alpha proteins, right. All alpha protein contains mainly alpha helices. You can see the dominated by alpha helices, right. So, more than 40% alpha helices and less beta strands, right. You can see an example, right. Here you can see several alpha helices. This is the structure for myoglobin, right. This structure for the myoglobin, right. It contains 8 alpha helices, but there are no beta strand here. So, you can see the structures. But all alpha proteins are getting popular because the first solved structure of globular proteins, right, which contains alpha helices, right. So, in this case it is popular, and if you see the membrane proteins, here also you can see that first solved membrane protein structures, right, that is photosynthetic reaction center, right, which also contains several alpha helices, right. So, alpha helices you can see the all alpha proteins are predominant, right. If you look at the all protein structures, you can see several structures they belong to all alpha class. Then the second class, likewise all alpha, some proteins they have more number of beta strands. Predominantly you can see the occurrence of beta strands, and these type of proteins are called all beta proteins. You can see the dominance of beta strands, right, you can see in the structures, right. So, you can see lot of beta strands, for example, more than 40% of beta strands, right, and less alpha helices, right. Okay, this is an example. Okay, there is a concatenaval A, so it contains several beta strands in this protein. So, one contains mainly alpha helices and the second class contains only beta strands. Now, the next possibility is the proteins contain both helices and strands. based on the location of these helices and strands, they are classified in two groups. One is alpha plus beta and another is alpha by beta, depending upon the location of this beta strand, alpha helix and beta strands, whether they are segregated separately or they are mixing with each other. So, one class is alpha plus beta proteins, right. In this case, helices and strands tend to segregate. You can see the presence of more than 15% alpha helices, right, and more than 10% beta strands, and the helices and strands, here you can see the helices and you can see strands, right. They are segregate from each other. This is an example for lysozyme, example for the alpha plus beta proteins. So, the fourth type of proteins, right, they are alpha by beta proteins, right. Here also you can see the helices and strands, but they mix each other. You can see the more than 15% helices and more than 10% beta strands, right. So, this is the numbers for understanding. It is not hard and fast rules to have this specific numbers, right. So, these helices and strands, right, which are mixed with each other, right. For example, this one team, right, contains 8 alpha helices and 8 beta strands. So, you can see alpha helices and beta strands, alpha helices, beta strands and so on, right. You can see the structures in the timbaral proteins. So, now how can we get the information whether a protein belongs to all alpha proteins or all beta or alpha plus beta or alpha by beta. So, to understand these characteristic features, right, there are several databases. The major one is the SCOP. So, to classify the proteins based on different structural classes. Not only the structural classes, then also have several sub classifications based on the how they fold, which family they belong to, what are super families, right, all the information they give. So, SCOP gives a comprehensive description of the structural and evolution relationship of the proteins of known structures. They started from the structures available in protein data bank and they classify the proteins, right, based on the information available in the structural data. So, they have the hierarchical levels. So, go with the family and then super family. For example, take a protein, right, first the small one they belong to some family, then different families they come close together and they have a super family, then different super families they belong to same fold. So, go with the fold and the different folds, right, they have the similar class, right. For example, the structural class all alpha, right. You can see 4 helical bundles fold and different types of helical folds, right. They are mainly a fold, these are the helical proteins having different folds, right. They belong together and under the class all alpha. So, for example, he give one example. So, here lysozyme, right, the family is lysozyme. Then you can see the fold, right, you can see the lysozyme like common alpha plus beta motif and the structural class alpha plus beta, right, and you can identify this protein in a 6-letter code. First 4 are the PDB code. Then how to represent the PDB ID? First one is the numeric, right, first numeric and then 3 letters
can be
that contain numeric or this is the any alphabets, right. For example, 1 L Z T M. So, you can this is always numeric and here these 3 either this is alphabets or it is numeric, right. So, these are the first 4 letters and if you have any chain information, for example, a protein contains different chains, right. For example, hemoglobin, how many chains in hemoglobin? 4 chains, right, 2 alpha and 2 beta. In this case 4 chains. For example, if I say ABCD, then you can see the chain information here. If it is A chain, it is A, B chain, it is B and so on. And the 6th one, you can see there are several domains. For example, if you have a large protein, it contains several domains, right, which are stable, right. If you know the domain information, then you can put the number 1, 2, 3, right, they have the domain information. This is how they represent the structure of any protein. So, this is the web server, right, the database of the SCOP. You can search the database, right, with any PDB ID, right, or the names. So, here you can the root is a SCOP, the protein belongs to alpha plus beta and you can see the fold is lysozyme like, right, and you have the super family and the protein name is lysozyme and the species Homo sapiens, it belongs to the human. Then they give the other entries which are relevant to the particular protein. For example, in this case it is lysozyme. So, what are other proteins, right, which is similar to lysozyme, you can give the details about the similar entries. So, here is a web server, the SCOP web server. So, here if you access this site, you can access this database and you can see the structural class information for several proteins. Similarly, there is another one server, right, this is also called kind of the database, right, they are called CATH. Here also they try to classify the structures based on the class. similar to scope, and the architecture and the topology and the superfamily. They use this word CATH, which represent class, A for architecture, T for topology, and H for homologous superfamily. So, what is C, what is A, what is T, and what is H? You see that class is the simplest level for different classes, just we discussed. What are the 4 different classes? Alpha, all alpha, all beta, alpha plus beta, and alpha beta, right. You can see the 4 different, this is the simplest way, right. And then go with the architecture. This summarizes the orientation of the structural units, like this is a barrels or sandwich and so on. Then the topology level, here you can see the sequential connectivity, how the members of this architecture, right, how they are connected. Sometimes you have the same architecture, but you have the different topologies, right, depending upon the alpha helicity beta strands, how they are connected with each other. Then the last one is the homologous superfamilies, that is H, or have similar structure and function. So, based on the connectivity and the proteins which have similar structure function or the orientation of secondary structure, right, they classify in different groups, right. So, this word they called as cath, C for class, A for architecture, T for topology, and H for homologous superfamilies. Okay, this is the database, the cath database. Okay, you can here for the same human lysozyme. So, its the class is mainly alpha here, and the architecture they put the number 1.10, this orthogonal bundle, and the lysozyme, right, and the superfamilies belongs to hydrolase. In the case of scope, right, what is the classification for the class? Right, lysozyme they put alpha plus beta, but here they put all alpha. Right. Most of the cases you can see the similar classifications, and some cases you can see the differences, right. I will explain why there is some difference, right, for the same protein in different databases. If you look at the several structures, almost all the structures in the protein data bank, and if you classify based on scope or the cath, most of the cases you can have the similar classification, right, that is not much different. In some cases it is possible to the different assignments in scope and cath, especially for the case of this alpha plus beta or alpha beta proteins, right. How this happens, right, how do you classify the all alpha? Mostly alpha helices. How do you classify alpha plus beta? It has both alpha helix and beta strands, right, depending upon this cutoff values, right. For example, if you take 10% or 15%, right, then you can say either they belong to all alpha or belong to another mixed class alpha plus beta or alpha beta, right. If the helical content is high, then you can see this can be all alpha proteins. Sometimes the helical content is high, but also it contains beta strands. In this case we have the conflict. If we consider the high helical content, right, in this case you can see this can be classified as all alpha. So, in this case human lysozyme, if you look into the contents, alpha helical structure is 31% and beta strand is 8%. So, it is high content of alpha, that more than 30%. So, classified as alpha all alpha protein, right, in cath. Due to the presence of helices and strands, more than 5% strand and more than 30% helix, right, it is classified as alpha plus beta in the case of scope. This happens only for few structures. So, in this case you need to check these databases, see whether there is consistency or not. Then based on your requirements you can classify this as all alpha or alpha plus beta. Then if you have the structures, right, you can get the structures from protein data bank, right, then you can derive several parameters. We can like from sequence we derived various factors. What are the various properties we derived from sequence?
Amino acid,
occurrence, composition, molecular weight, average property values, and then hydrophobicity profile, and we can do the dipeptide composition, and we can do the alignment, multiple sequence alignment, conservation, right, several features you can do it. Likewise, if we have the 3D structures, we can do better, because 3D structures contain more information than amino acid sequences. So, we can derive various parameters, right, and the important aspect is whatever the parameter we derive from the structures, they have some implications, they have some applications to understand the structure or function or anything related to the diseases. I will explain some of the features. For example, contact maps, right, this is the simplest one we can construct from protein 3D structures, and accessible surface area, contact order, right, depending upon the how the two residues are contact in protein structures, and long range order, that depending upon the contacts which are close in space, but they are far in the sequence level, and some cases some residues have more number of contacts. some case less number of contacts, right, some case more number of contacts. For example, in a class, for example, if a class representative, right, we contains large more number of contacts, right, some students they have less number of contacts, right. So, then these contacts are important, right. So, the residues which have more number of contacts they have higher influence, right. This is the principle of multiple contact index. Then we can derive other factors like hydrophobicity, buriedness, transfer free energy, how the accessibility is reduced from the unfolded state to goes to folded state, how to get the different interactions, cation pi interactions, electrostatic interactions, hydrophobic interactions and so on. So, we will explain some of the parameters, right, and derive how to do it. So, first one is the simplest one we can obtain from the 3D structures is the contact maps, right. So, what is the contact map? It simply represents the distance between different residues in protein structures. So, we have 3D structures, the atoms and the residues are located with the XYZ coordinates, right, and in the contact map you can see how the residue 1 and 2 are in contact with each other, 1 and 5 are in contact or not. So, the representation of the 3D structures into 2D graph. For example, x axis if you have the amino acid sequence, right. So, amino acid sequence here and y axis you have the amino acid sequence, say 1, 2, 3 like that, right. Now, the question is whether these residues are in contact or not. If they are in contact you put a dot, if no contact leave it, then you will get a matrix, right, we will show you whether which residues are in contact, right. I will show you some examples. So, two residues I and j, one the residue I, second another residue j, right. If it is equal, if this matrix is 1, if the residues are closer than any specific threshold, right, any distance. In this case we need the information regarding the distance, right, and then getting the distance which atoms we need to consider. These are two different aspects we need to construct a contact map. So, if you look at this is the example for the contact map just I showed earlier. So, here you have the amino acid sequence x axis, y axis amino acid sequence, right, and you can put a dot if I and j are closer in a specific threshold, right, otherwise it is a blank. If you see this graph, what you can infer from this graph? So, diagonals you can see it is always present. What does it mean?
Nearby residues are
Nearby residues, right, because they are near the van der Waals contacts. So, in this case 1 and 2, 1 and 3, like 2 and 3, 2 and 1. So, they are always in contact, right. So, this way it is in the diagonal you can see always present. And if you close to the diagonal, some cases you can see some residues are present, right, some cases no, in this case there is no, this case there is no, right, most of the case it is yes. But if you look at the specific cases, for example, here it is the very far, in this case it is around 10, this around 300. These residues they are close in space, but they are far away in the sequence, right. I will explain the details now. When you make the contact, right, as I discussed earlier, there are two different aspects. One is we need to fix the distance, second one we need to fix the threshold. If you take the atoms, there are various ways to define this. Either you can consider C alpha atoms, that is the simplest one, take all the, consider only C alpha atoms and see the distance, or you can say C beta atoms, right, because C beta atoms you can see the interior of the protein, right. This way many researchers they use C beta atoms. So, can we use C beta atoms for all the residues?
No, except like
No, right, because one residue, right, for glycine does not have the C beta. So, in this case they use alpha and all other residues they beat beta, right, they represent better than the C alpha. So, they use C beta atoms, or you can ask any atoms. For example, residue 5 and residue 10 they are in contact or not, you can see any of the heavy atoms which are within the specific distance, or you can see centroid, any atom any residue you can get a centroid XYZ coordinates, right. Now all the residues, right, are represented by centroids, then you can calculate the distance and then you can see the cutoff, right. So, there are various ways you can consider atoms, either C alpha or you can get C beta or all any heavy atoms, or you can use centroid. Then the distance, which distance we need to consider, 4, 5, 6, which distance you want to consider depending upon the atoms you consider. For example, if you use C alpha or C beta there you can use the distance of 6 to 12 angstrom, right, because we consider only one atom. In the case if you have all atoms, then you can reduce, or you go up with 4 angstrom or 5 angstrom, otherwise you will get more number of contacts, right. So, depending upon the atoms you use, either C alpha or C beta or all atoms, you can define a threshold. So, now here this is the coordinates. So, this is the residue name. What are the coordinates? Here, right, XYZ coordinates. What is the next one? Occupancy. This one? B factor, right, okay. So, you have the XYZ coordinates, right. Any protein structure if you go, I showed earlier about the protein data bank, right, one example. So, you will get the same the level of this representation, you can see XYZ coordinates. You can use these coordinates to construct contact maps, right. For example, I show the coordinates, these X, Y, Z coordinates. So, this is the C alpha atoms, because I extract the C alpha atoms from here, right, this is 36.942, okay, see the C alpha atoms, right. So, here this is the coordinate 36 minus 23 and minus 8, right. I extract all the C alpha atoms. In this case, which distance is better to define? 6 to 12 angstrom, 6 to 14 angstrom. So, I use the, let us use the distance of 8 angstrom, right, because these are the C alpha atoms, and you can construct maps. How to construct a contact map? Right, first this is what is the x axis, this is a sequence, right, this is a sequence here. Here also you have the sequence. So, now we have the residues, right, you can see this is the 1 is methionine, 2 is asparagine, 3 is isoleucine, right, we have the residues, or I can put the numbers, okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, take 1 and 2 are in contact, yes, right, 1 and 3, yes, 1 and 4, about 1 and 5,
1 and 4 not in contact.
1 and 5,
Not in contact.
1 and 5 also no,
Uh,
here 3 9, 36 plus 9 equal to 45, 16,
9,
okay, maybe in contact, let us see,
4,
about 1 and 6,
1 and 6, 9.
Not
In contact.
in contact, 1 and 7, no,
No.
1 and 8,
1
No.
and 8, no.
1 and 9,
No.
no, 1 and 10, no, okay, 12 and 20 is already 7, right. No, okay, then the 2 and 3,
Yes.
yes, 2 and 4,
7. Yes.
2 and 5,
4.
yeah, 2 and 5, yes,
Yes.
2 and 6,
Yes.
yes, 2 and 7, 2 and 7, no, right,
No.
2 and 8,
No.
2 and 9,
No, sir.
no, 2 and 10, no, 34, 43, so this no. Then we'll go to 3 and 4, yes,
Yes.
3 and 5, yes, 3 and 6, yes, 3 and 7, yes, 3 and 8,
No.
oh, no, 3 and 9, no, 3 and 10, no. Then 4 and 5,
Yes.
yes, 4 and 6, x, 4 and 7,
Yes.
4 and 7, okay, yes, 4 and 8, probably yes, 4 and 9, maybe no, right, yeah, 4 and 10, no, right, then 5 and 6, yes, 5 and 7, yes, 5 and 8, yes, 5 and 9, yes, 5 and 10,
Yes.
maybe no, then 6 and 7, yes, 6 and 8, 6 and 9, no, 36, 13 and 43,
Yes.
yes,
Yes.
so 6 and 10, 4, yes,
Yes.
okay, I'll put yes, then 7 and 8, yes, 7 and 9, yes, 7 and 10, yeah, yes, then 8 and 9, 8 and 10, 9, 9 and 10, right. So, you can construct the matrix. So, we will get the symmetrical matrix or the non-symmetric matrix, this one
We'll get
asymmetric, right. Why it is symmetric?
we haven't drawn the set.
Yeah, we did not draw another set, because if 2 and 3 are in contact, then 3 and 2 are also in contact, right, not like the amino acid sequence. There you talk about the neighboring residues. So, in this case you do not get symmetric matrix, right, but here if 2 residues are in contact, right, 2 and 3 in contact, 3 and 2 are also in contact. So, in this case you can get the symmetric matrix. So, here you can see the diagonal. From this one you can easily say that the residues are influenced with the short range contacts, because the residues which are very close. Then some residues, right, you can see even near the diagonal 2-3 residues which are far away, they are also contributing, right, or they are also interacting in protein structures, right. And here we consider only 10 residues, this is why we will show, we could see the long range contacts, but in this figure, right, we can say the long range contact for this. So, depending upon these contacts, right, based on the space and how they are located in the sequence, we can classify into different types of interactions, right, different types of contacts, whether these contacts are short range, that means short in terms of sequence, right, because we fix a space, because when you construct the map, the space is fixed, any distance 6 angstrom or 7 angstrom or 8 angstrom we fixed. Now, the difference is only at the sequence level. So, the sequence level we see whether they are very close in sequence, right, and how far they are apart in the sequence. For example, if we see this one, right, the T is the central one and the radius is 8 angstrom, we construct a sphere and there are several residues. Now, these residues are located in the sequence which I give below, right. You can see this the T is central, we can see the 3 here and how they are distributed along the sequence. Based on the distribution of the residues along the sequence, we can classify into different types. One, we can see short range contacts, that we want to see the residues which are close to the central residue, either +2, +1 or +2. Almost all residues they have the short range contacts, right. So, if we take any residue, how many short range contacts each residue will have?
3, 4.
4 residues, right. For example, take 5, for example, residue number 5. So, if we residue number 5, then you can see 4, 3, 6, 7. So, 4 contacts. Then you can see whether any residue, the residues they have contact with at least +or -3 or +or -4 residues. In this case, you can see some medium range contacts. We use 3 or 4 because we need to represent the type of alpha helices, right. How the alpha helices are formed, what is the range? I and I plus 4, right. In this case, you can see it will come within this range, and you can see long range contacts if it is more than 4, but this limit is so far because you can see they are very large. So, you can see even 4 we can divide into small bins, 4 to 10, 11 to 20 and so on, and then see which range you can see the contacts in different types of structural classes. So, here I show a figure, this is the contact of T152 of lysozyme, there is a real contacts. If you look into the PDB structure and take the 319 and if you get the residues which are within the limit of 8 angstrom, you can get this figure. If you see this one, we can define the short, medium and long range contacts. Some residues, for example, F153 and 151, they are just neighboring residues. These residues form short range contacts, and some cases, for example, 156 and 155, they are 3 to 4 residues far apart. So, they make medium range contacts, and some residues which are far away in the sequence, but they are close in space. For example, 152 and A98, right. What is the distance in the sequence level? 54 residues, right. So, they are far away in the sequence. So, these residues contribute long range contacts, right. So, if you take a structures, then you can see the contacts based on the short range, medium range and long range. You can also interpret in terms of alpha helices and beta strands. The alpha helices you can see dominated mainly by the medium range interactions, and the beta strands which are dominated by long range interactions. Why? Because of the hydrogen bonding patterns, right, in the alpha helices and the beta strands, right. So, now if you see this one, diagonal ones, they are mainly, these are the short range contacts, and you can see the residues very close by the diagonal, these are medium range and here you can see they are long range. And if you see the local secondary structures, for example, if you see these are the beta strands, right, you can see the beta strands here because there are many long range contacts, and several cases these are mainly alpha helices. And if you see the distribution of secondary structures and the contacts, easily you can say that which region belongs to helices, which region belongs to beta strands based on these patterns. The long range contacts mainly they are far away from the diagonal. So, now I show the data for the 4 structural classes. We discussed 4 structural classes, right. What are the 4 structural classes we discussed? All alpha, all beta, alpha plus beta and alpha beta. So, if you see the all alpha class is dominated with helices, right. Helices are well dominated with medium range interactions, right. So, in this case if you see we have different residue intervals, medium range 3 to 4, and then we can see 1 or 2 residues apart 5 or 6. So, you can see more than 25% of these helices you can see mainly at this level 4 to 10 interval, and you can see it is very less for the different cases, it is going down, right. There are the contacts from the long range is very less in case of all alpha proteins. But if you look at the all beta proteins, right, this is the all beta proteins, right. Okay, this is all beta proteins. So, here all beta proteins you can see the range is 11 to 20, because mainly these antiparallel beta strands, okay, they have the hydrogen bonding pattern with respect to more than 10 residues. So, you can see 11 to 20 residues, that is the dominance in case of all beta proteins. But look into alpha plus beta proteins, right, that is in between, right. You can see both the cases, this is the all alpha, is all beta, you can see that is in between that. Here also all beta, here is all alpha, in between all alpha and all beta. But look at the alpha by beta proteins. So, what is a specialty in alpha by beta proteins? Alternated, one alpha, one beta. In this case, if you take 2 beta or if you take 2 alpha, the distance will be more, right. If you take the beta strands, right. So, beta, alpha and beta. So, in between 1 alpha, it crosses more than 10-15 residues. This is the reason if you take this type of proteins, right, we can see this is dominant in the 21 to 30 range, because several timbarrel proteins, right, in this if you see the hydrogen bonding pattern, they are between 21 to 30, this way it is dominant in 21 to 30 range. So, that makes sense, and if you see the structures you can relate the secondary structures, structure class as well as the number of contacts in protein structures. This is another, I show example, this is all alpha, this is all beta, right. If you look at these figures and based on the number of contacts, mainly the long range contacts, right. So, which one has more number of long range contacts? All beta is expected, right. So, you can see more number there up to 13 contacts, but here it is very less, and most of the cases it is 0. Many cases you can see 0, there is no long range contact in the case of all alpha proteins, but here there is very less, most of them are having more than 6 long range contacts. What do you expect for the alpha plus beta and alpha by beta, right. Alpha plus beta if you take, you can see the mixed combination of these two. One part it should be less number of long range contacts, and another part you can see the more number of long range contacts. You can see this, right. If you see here, okay, here you have more number of long range contacts, here you have less number of long range contacts. Here most of the cases they have 0 long range contacts, that is fine, right, you can understand. So, then the alpha by beta you can see the zigzag, right, here some high and low, that is you can see the patterns. So, in terms of these contacts you can easily understand different structure classes and how they interact and how they make the patterns, then easily you can see the different functional aspects, right. This is the contact they will make and how we can superimpose the different structures and to understand the different functions. So, now we have the contact maps. To construct the contact map, what are the different information do you need?
Coordinates.
We need the coordinates, and we need to consider the distance plus the atoms, right, depending upon the atom type, right, you can fix a distance and you can see if this is in contact, you can put a dot and you can see construct the contact maps. That is simply the representation, right, of the 3D structures in the 2D space, right. Then you can understand how the residues they contact with each other different structure classes.